Thank you, my esteemed colleagues, for the privilege of addressing you at this 2015 Toronto Biomarkers Conference. I would have loved to have returned to the country of my birth to meet you all and to learn from you, but at this moment I am on vacation. Now, I could deliver the whole talk from behind this podium, but in the interests of keeping you awake, instead we'll be doing it from... We're on location atop the parking garage across from my office right over there. Today in this brief talk, I'd like to explain to you why I believe telomeres are the ultimate biomarker for aging and disease. And to do that, I'll be explaining my stem cell theory of aging and disease. They say it's good to open with a joke, so this ad from the UK for telomere testing is my joke. It's really kind of a sick joke, actually. Do you remember the movie Blade Runner, where the android knew up to the very moment when he was going to be dead? Well, telomere testing is decidedly not that kind of thing. Companies that recommend telomere testing have an interest in oversimplifying things, but the fact is, it's a little more complicated than that. There is no app that can tell you your exact date of death, but there is an app that can help you live forever. That app is called Telomeres, and it's contained in every single stem cell in your body. That's what we'll be discussing at the Thomas Noguchi Sculpture Garden, right over there behind that building. But first, let's go to the Rose Hill Cemetery, where my father's father, and my mother's mother are all laid to rest. It's funny, they always try and get me to buy a little pied-à-terre over there, just like I rent a space in this parking garage. But what if this park won't need to park there at Rose Hill Cemetery until the 24th century? I'll explain. We're here at the beautiful Rose Hill Cemetery to illustrate a point. The loved ones of the people interred here probably thought they all died of something different. But consider these articles, thousands of articles, all showing that telomere dysfunction is related to the diseases that took them. In this cemetery, my mother's mother, my father's father, and my own father was laid to rest in 2006 after he lost his battle with brain cancer. So, how long do I really think I can live? Well, here's a chart of my telomere length since 2007. As you can plainly see, they've only gotten longer, so I may already be immortal. But even if I were to stop taking telomerase activators and age at the normal rate, this suggests I may have 250 years to live. Given that fact, I think I'll hold off on buying a plot for now. Back in 2006, my father was about to succumb to his brain cancer. Dad was a general practitioner, and he served in Saskatchewan for many years. He knew what was happening, but neither of us as physicians understood why, the why of cancer. When I Googled or looked up why people get old and sick, really only one explanation made sense to me, and that holds true to this day. That is a telomere theory of aging. As you all know, the ends of the chromosomes have telomeres on them. These telomeres shorten every time a cell divides. Now, when the cell thinks that they're too short, it can't tell the difference between that and a clean break, say from an X-ray. So what it does is it heals ends together. That causes double chromosomes like trapeze artists connected at the hands. And the next time cell division occurs, you'll get the centromeres and the telomeres pulled apart, but there'll be tears and mutations in the cell. Enough of these tears and mutations leads to the cell recognizing that it's damaged, and it causes the cell to go into crisis or apoptosis. Now, if I was to take a cell from your body and put it in a dish, it would only replicate 50 to 70 times because of this end replication, or this replicative senescence problem. That's the Hayflick limit. In contrast, we know that cells over the course of a lifetime only age about 50 to 100 base pairs a year. How is that possible? Well, that's because our stem cells are constantly regenerating their telomeres, just like this stem is constantly regenerating itself. These dead pine needles have no telomerase activity, whereas the further you go to the trunk and the root, the more stem activity there is, the more telomerase activity is. And that's why stem cells are immortal, and that's what leads us to my theory of aging and disease. We're standing in the sculpture garden by Osamu Noguchi called California Scenario. In 1936, a young Osamu Noguchi asked Buckminster Fuller to explain E equals mc squared, and Buckminster Fuller telegraphed back this interesting message. It's funny, Einstein spent his entire life trying to find a theory to unify everything, or a grand unified theory that would explain how electromagnetism, gravitation, and the strong and weak nuclear forces were all united. Well, we're here to explain my theory, my grand unified theory of aging and disease. So let's do that right now. Axiom number 1, telomeres always shorten when the cells replicate. It's like water always flows downhill. Axiom 2, non-stem cells do not activate telomerase. Therefore, they do not live forever. These dead pine needles were not meant to live forever. Axiom number 3, a daughter cell can only be as healthy as its most recent mother, and it can only be as young as its most recent mother. This statue is called Spirit of the Lima Bean, and it should be obvious that a lima bean can only be as healthy as the lima plant from which it came. Axiom number 4, stem cells use telomerase to restore the length of their telomeres, thereby staying young. Just like somewhere under this pyramid, there's a pump that needs to restore the water back up to the origin over there. So, my hypothesis is that telomere shortening in your stem cells and the accumulated damage is what causes aging. It follows that if we can prevent or reverse this telomere damage, then we can prevent and reverse aging itself. We're standing right in front of the El Toro Y, where the 5 and the 405 freeways meet in Southern California. It's 26 lanes wide, and the reason we're here is because when you look at a normogram for telomere lengths from any study or any lab, they're going to be 26 lanes wide. In other words, you cannot predict someone's chronological age based on their telomere age. Having said that, telomere measurements are very precise and they're very reproducible. So it's a great way to personally benchmark for individuals. Now, if you consider this chart from the Kaiser study, 100,000 patients, what you can clearly see is that the trend is not your friend. There are 3 methods to measuring telomeres. Some would say they're like comparing apples to oranges, but I prefer to compare them to different types of orange juice. Let me explain. The first is the gold standard Southern blot, where you take the DNA, you cut it up with restriction enzymes, and then run it on a gel electrophoresis. It's very precise, but it's very labor intensive, and hence not available for public use. Just like this juice, the gold standard Southern blot comes with a little extra, be it pulp or calcium. So in that measurement, you get about 4,000 base pairs of sub-telomeric measurement added on. The most commonly available test is quantitative PCR, which would be sort of like juice from concentrate. Why? It's easy to make, you don't need fresh cells, and it's very affordable. The problem with quantitative PCR is that it has a little bit of inter-user variability, and so you have to always check and make sure that the tests don't vary too much. It said that probably about 7% variance exists between the same blood sample run on different samples of quantitative PCR. The last method is the fresh squeeze method. That's high throughput quantitative FISH. With FISH, or fluorescent in situ hybridization, you actually need live cells and you have to paint the DNA, then measure the amount of light that is painted on those telomeres. I prefer this method because it gives you a direct measurement of all the ends, and in one case you can actually get a percentage of critically shortened or damaged telomeres. We're here at the Santa Ana Zoo to talk about animal models of aging. Many people believe that since laboratory mice have long telomeres and they still age, this proves that telomeres are not involved with aging. In fact, mice are inbred mutants, and they have freakishly long telomeres in the lab. Also, they can't respond to oxidative stress in the same way that humans can, and therefore have very high rates of genetic mutation. Now, this is a gibbon behind me, and our closest relative, as we know, is a chimpanzee. Chimpanzees in captivity conditions like this only live on average to their teens when you include infant mortality. Why is that? Well, perhaps it's related to the stress of being in captivity. Dr. Jane Goodall, famous primatologist, says that in nature, chimps can live into their 50s and 60s. What's the difference? Well, maybe chimpanzees with their twice the telomere length, yet twice the telomere erosion, could live as long as we do if they only had Amazon.com, better public health and clean water, and hot yoga classes. We don't know. I believe that AIDS is a form of aging, and aging is a form of AIDS. They are both each other's cause and each other's shadow. For example, chronic infection throughout our lives with CMV and other herpes viruses grinds down the system. It's that overuse, overcopying, and replicative senescence that causes us to lose our immune system and to age. Why do men like Jack LaLanne, who are physically fit, or Nelson Mandela, who are spiritually advanced, why do they die of the common cold? It's because they ran out of blank keys. Their naive T cells were used up. Consider individuals with trisomy 21 or Down syndrome. They age at 3 times the normal rate because they have 3 times faster telomere attrition due to their dysfunctional T cells. I'd like to take this moment to talk about the idea that telomerase activation causes cancer, as some of the experts in this field may have suggested, despite recommending telomerase activation by natural things such as meditation. I believe the greatest contribution is the landmark study in the Journal of the American Medical Association in 2010 by Willett et al. What they showed was that there was a dose-dependent relationship between telomere shortening and both the incidence and mortality from all causes of cancer. Another interesting study out of Northwestern University by Howe et al. In 2015 showed that this is happening in 2 phases. They looked at men from the Veterans Affairs and looked at their charts. They found that telomere erosion happened quickly up until about 3 to 4 years before the clinical cancer manifested. This is in keeping with my stem cell theory of aging disease. Rapid telomere shortening allows the ends to cause fusion, which then cause mutation in that cell line. The crisis stage is when the stem cell is damaged and it's negotiating with itself, its neighbors, and the police or the immune system as to whether it will manifest as a clinical cancer. I believe that we are destined to get every single cancer if we can survive long enough. It's like having a car for 1,000,000 miles. You don't expect to have any of the original parts. Same goes for clinical cancer. Now, I subscribe to a cancer stem cell theory, which is that cancer does not, as we were perhaps taught in medical school, arise from differentiated run-of-the-mill cells. Rather, it primarily affects stem cells. In other words, you have an already immortalized stem cell, which then mutates. It doesn't have to learn how to immortalize through telomerase. It retains that capacity. It's like saying Jimmy Olsen doesn't learn how to fly, he's Jimmy Olsen. But Clark Kent, already being immortal and flying, can become Bizarro Superman. So, now we understand that the excessive copying, the replicative senescence happening in your stem cells is what's causing the cancer. And all those risk factors that we associated epidemiologically speaking are really just proxies for accelerated cell copying without enough time to regrow the telomeres. For example, smoking, coal mining, in the lungs. The lungs have to turn over too fast, the stem cells can't keep up. Drinking in the esophagus, in the liver, same thing. Unopposed estrogen for the breasts and for the uterus. Unopposed ovulation, overuse of the ovaries for ovarian cancer, and excessive stimulation of the prostate by high testosterone levels. One of my biggest pet peeves in telomere research, and there are over 15,000 articles in the literature, is that most of them assume that blood cells and their telomere lengths will represent aging in other organs. The good studies study the organ of interest. For example, If I were to measure the tire pressure just because it's easy, will that tell me what's going on with the carburetor, the brakes, the electrical? Of course not. If this car came from Colorado, the underbody might be shot from road salt. If that car came from San Francisco, the brakes might be shot. Same thing with our body parts. Study the organ of interest and its telomere lengths. So that's really most of what I wanted to tell you about telomeres as a biomarker of aging and disease. Now it's up to you if you want to do the science that will help validate this grand unified theory. They used to say that there's 8 million stories in the naked city. Well, what if there's only just one? My name is Dr. Ed Park, and it's been my great privilege to present to you at this 2015 Toronto Biomarkers Conference. Thank you for your attention, and I hope with your efforts we can keep parked in places like this and not the other place. I wish that you and the telomeres in your stem cells will live long and prosper.